um, what are the general properties of these viruses? So, like I said, they're broadly distributed among humans, other mammals, and birds. They're thought to cause only respiratory and gastrointestinal diseases in domestic animals and common colds in humans until SARS showed up. These are extremely large viruses. Their genome are about 27 to 30 kb, largest for RNA viruses. They're single-stranded, non-segmented, positive sense RNA, which means that once they get into the cell, they will be translated because their genome, these genomes resemble cellular mRNAs with a 5' cap and 3' poly A tail. And like I said, the cool feature of coronavirus is they're the largest genome of any RNA virus, which also makes it hard when you think about generating infectious clone and replicon systems. So one reason why it's very hard, it's very challenging to eradicate coronaviruses from humans is because they're found in so many different animal hosts, as I mentioned earlier. And we know that most of these viruses are found in bats as the reservoirs. And what is thought is that the virus would jump from the reservoir host into intermediate hosts, as you can see here, a wide range of species, where the virus would adapt to cross-species transmit into humans. And so you see here that SARS-CoV-2 went through the Himalayan palm civet cats, and MERS went through camels. And SARS-CoV-2, the verdict is still out, but it's likely to have gone through pangolins. And the reason why I'll get to in a second is the sequence identity. And so we know now that SARS-CoV-2 is about 80% identical to SARS-CoV, and it's only 50% identical to MERS-CoV. So it's very distinct virus. It's a distinct virus from the other coronaviruses that have caused outbreaks in humans before. We have now found, groups have now found a bat coronavirus, this strain called RATG13 that shares very high sequence identity to SARS-CoV-2 at 96% on the whole genome level. And so it's basically suggesting that it is possible that the virus could directly jump from bats. But what is interesting is that coronaviruses are also found in pangolins, and they share about 91%, you know, there's a range, but around 91% identity to SARS-CoV-2, not as similar as SARS-CoV-2 is to the bat coronavirus. However, when different groups looked closer at the receptor binding domain of the pangolin coronavirus, what is really striking is that it's almost identical between the pangolin coronavirus and the SARS-CoV-2, except one mutation. And so there are certain theories or hypotheses that have arisen that is suggesting that perhaps SARS-CoV-2 might came from a recombination event between the bat coronavirus and the pangolin coronavirus. So because I want to sort of get into the details of the basic replication strategies of coronaviruses in general today, I thought it would be good to first give you an overview on the viral life cycle. Basically, what does the virus need to do when it gets into the cell in order to replicate and also generate more progeny viruses. And so for coronaviruses, it binds to the receptor on the cell surface, and usually it undergoes receptor-mediated endocytosis and fusion, either in the endosome or direct fusion at the plasma membrane, depending on what coronaviruses you're looking at. In terms of SARS and SARS-2, the receptor has been shown to be angiotensin converting enzyme 2, ACE2. It's interesting because this receptor is found in both the lung and the gut, and so there's also gut symptoms that have been reported for SARS-CoV outbreak before. Once the virus get into the cell and releases viral genome, the first thing it needs to do is to translate replication complex proteins, and it's in the form of a giant polyprotein called ORF1. It's the replicase ORF1 protein. And once the replication complex is being established, then now the replicase can make more mRNAs. And so the transcription basically happens on virus-induced membranes. And this is characteristic of a lot of RNA viruses that set up their replication complex on membrane-bound compartments in the cell. And so from there, the virus makes this nested set of mRNAs, which I get to. It's a very cool and cool mechanism that is not completely characterized. And from these nested mRNAs, proteins, structural proteins can be translated. At the same time, the full-length genome is also being replicated. And so when the structural proteins get translated, they can then form the virions that package the newly synthesized RNA. And this nuclear capsid protein coded RNA basically gets, it basically traffics through the ER trafficking pathway, and it butts out and it gets released eventually to infect new cells. And so you can imagine that in order for the virus to be successful and make more of itself, it needs to intersect with many different pathways in the host cell. And this is actually an area that my lab actively studies. So you can imagine there are two ways, right? The virus first needs to co-opt cellular machinery for replication. So those are considered pro-viral factors. But the virus also need to antagonize the host immune responses, which are activated once the cell senses the viral nucleic acid, for example, the double-strand RNA, and so that it can suppress that immune response.
Melody,
so, yes.
Question here.
Yes.
So in this slide of the life cycle, where does chloroquine come in? I don't see any acidification of endosomes here.
Yes, this one is simplified because depending on what coronavirus you're looking at, the virus either can fuse directly at the plasma membrane or, like you said, fusing with the endosomal membrane. So if the endosomal fusion is shown in this schematic, that's where the hydroxychloroquine would be acting at, I believe.
So presumably that's true for SARS-CoV-2, right?
Yes, and actually
sensitive in vitro to chloroquine.
Right. So actually, we've been collaborating with Vythe and in the BSL-3 studies, and we see very clearly that hydroxychloroquine is amazing positive control in vitro on cancer cell lines. However, you have to pre-treat the cells at least an hour before the infection or addition of the virus with the hydroxychloroquine to see this very dramatic inhibition of our replication.
Okay. Mm-hmm.
Great, thank you. Any other questions? Okay. All right, and so, so I'm just showcasing a few studies here because obviously there's so much literature out there that is, you can find on bioRxiv. But sort of give you an example of both the proviral versus the antiviral side of things I just mentioned. In this host interaction, host-virus interaction picture, this study, this preprint shown here by Wei et al., basically they have done a genome-wide CRISPR screen, but using actually a library from African green monkey cell line. Where they identify cellular processes that modulate viral infection. And so shown here are both guide sgRNAs that are positively selected, which usually means that they are proviral because knocking down these genes protect the cells from virus-induced cytotoxicity. whereas the negatively selected processes shown in the blue are potentially antiviral genes that are basically their guides are being depleted in the surviving cells. And so we can see here that a few different processes, these are only showing the top three processes for both potential pro and antiviral functions. And you can see a lot of these are important for chromatin accessibility, gene expression, transcription. And what is interesting is that viral translation components are found in both populations. On the other hand, I mentioned with the host innate immune response, that's also one of my lab studies, which is type 1 interferon. And that is the first line of defense against the virus when the cell first sees a virus. And so you can see here what is really interesting is that when this group, again, about a preprint, when they looked at SARS and SARS-CoV-2 replication in viral cells, the kinetics is very similar. However, when they treated the cells before infection with type 1 interferon, they found that SARS-CoV-2 is hugely more sensitive to interferon pretreatment than SARS. And so that brings up a really interesting question, because I'll get to that also in a little bit in a later slide, is that because coronavirus is so big, it can encode so many more genes than other RNA viruses. It also has a lot of bells and whistles to antagonize the host innate immune response. And so shown here, a comparison done by this particular group, where they compare sequence identity between SARS-2 bat coronaviruses and SARS-2 for all these individual viral genes that have been shown to have interferon, some kind of interferon innate immune antagonism activities. And what is really interesting is that for some of these genes, NSP3, which I will also touch on later, OR3B and OR6, there seems to be a lot more differences found on the sequence level between SARS-2 and SARS, suggesting that these could be potential viral genes that have somewhat attenuated or defective activities in terms of counteracting the host innate immune response. And I just want to point out TR here just means that the OR3B gene is truncated for SARS-2. Okay, so going back to the replication strategy of the virus, I mentioned earlier that before the virus can replicate its genome, the first thing it needs to do is to make the replicase complex. And so from the viral genome, the replicase ORF1A and 1B are the only two proteins, and they're polyproteins that are translated from the genome. And I'll get to it, how it's done. The rest of the genome encodes structural components, and these are translated later after the nested mRNAs are being generated. And you can imagine that from the virus perspective, this is a very it's smart, because it's a timed expression of different components of the virus that are required at different times of the viral replication. And so you can see here that for the replicase 1A and 1B, what happened is that most of the time the ribosome actually terminates at this stop codon. And so there's a stop codon at the end of replicase 1A. What happened is that this stop codon is actually part of this RNA pseudonon. It's a secondary structure. However, once in a while, the ribosome would actually slip, because the sequence here is very U-rich, and that usually causes slippery to happen for the ribosome. And so instead of getting to the stop codon here, shown here, basically the ribosome will slip, and that allows frame shifting to happen. So it's very smart. So that this is how the virus can basically controls it, so that only some of the times now the ribosome slips back, and now it goes to a different frame. And now it generates the entire polyprotein called the polyprotein 1AB. But most of the time, it only generates the polyprotein 1A. And I forgot to mention that usually the RNA structure also needs to be unwinded so that basically the ribosome can go through. And so here is, I'm showing you the polyprotein schematic for both replicase 1A and 1B. And the little lollipop and the arrow signs here are basically telling you which of the two viral proteases is actually targeting these cleavage sites. And so, and it's a very complicated slide. I really just want to point out a few things because there are just so many viral proteins. But what I wanted to point out is that NSP3 here contains the papain-like protease. And so the sites are targeted by the lollipop signs here. So you can see these are NSP3 responsible cleavages. And then the rest of the sites, the arrow signs are NSP5 responsible cleavages. And that's the main protease, or the M-pro. And so once the polyproteins are being translated, as I mentioned in the last slide, and proteolytically processed by the two proteases of the virus, now these proteins are basically individually expressed. And among them, NSP12 is the RNA-dependent RNA polymerase, the RDRP. And I'll get to it at the end of the talk. How does coronavirus deal with the issue that it has such a large viral RNA genome? And the magic here is it encodes a non-structural protein 14 that actually has proofreading activity. So this is the exonuclease, also called exonuclease. But obviously, it also interacts with other viral proteins, which I will not mention today. So I mentioned earlier with the bells and whistles that the virus encodes to antagonize the innate immune response, just to highlight some of them that are really cool mechanisms. For example, NSP1 of coronaviruses are interferon antagonists, where this protein actually can promote host mRNA degradation, including mRNA of the interferon gene. NSP3, it's considered a Swiss Army knife in the field, because you can imagine just how complex this protein is with ubiquitin-like domain, macrodomain, and protease domain, and even deubiquitinase activity. And so this protein has been shown to encode all kinds of antagonism activities, including interferon, NF-kappa B, and antiviral PARP, poly-ADP ribose polymerase activity. And so how does, I guess, getting back to, now the next step in the replication cycle is that now that all these proteins are being expressed, how does the virus actually replicate its genome and also make its mRNAs? And so what is really interesting about coronaviruses, getting back to the nested aspect, is that all the mRNAs are subgenomic. So they are shorter than the full-length genome. This is the full-length genome, and these are all the nested subgenomic mRNAs. And these encode the structural components of the virus, which are basically in the last third of the genome. And what is really curious at first to virologists is that all these mRNAs share the same sequences on the 5' end and the 3' ends. So how does it do that? And so the next slide could potentially confuse the heck out of everyone, but I'm gonna go over it because I think it's such a cool characteristic of coronavirus, and I call it sort of the martial arts feature of coronavirus, because it can jump through the sequence. And so the way the virus does it is it goes through discontinuous transcription mechanism. And so shown here is that for the on top is that the viral genome actually has these conserved sequence regions that are called TRS. So these are the transcription regulating sequences. And they're conserved between the leader and the body. And you can see in the previous slide that all of these mRNAs, including the full length, contains the leader sequence, the leader TRS. But then the rest of the genome, in front of each of the structural gene, it also has a body transcription regulating sequence. And so how the virus does this is that when the RNA, the viral polymerase is extending, is making its negative sense RNA from the positive sense RNA. When it gets to this conserved sequence, the TRS, right, in the body, TRS body sequence, what it does is that it gets to here, right, it elongates until it gets here. And what happened is that it could either choose to keep going and make the full length, or it can jump basically to the beginning. So if it wants to make the full length, it would basically keep going, and then it could jumps to the beginning of the this conserved motif because all these RNAs are also interacting with each other and the RNA-dependent RNA polymerase. Or it could basically stop at each of these conserved sequences and then quit, basically, by jumping over to the end of this, the 5' end of the original RNA genome. And so at the end, what happened is that it would basically make this nested set of negative strand RNA, which then will serve as a template for mRNA synthesis. So the idea is that every time when it gets to the conserved sequence, it could either stop there and then jump to the 5' end of the original viral genome and then finishes basically the synthesis, or it can keep going to the next one or to the next one. It is not clear what are all the factors that determine the choice of, for example, the ratio of these different transcripts and when does it decide to keep going or when does it decide to stop. But potentially it has to do with also proteins interacting with these conserved sequences in the RNA. The other thing I want to point out is that this, you can imagine, also create a mechanism for RNA recombination when you have basically two different strands of RNA genome. Okay, so I probably confused all of you guys, but in the interest of time, I will jump to sort of the last part where I want to mention how about the large genome size of coronaviruses. How does it deal with it? And so here I'm showing you a graph that basically looks at the genome sizes of all single-strand RNA positive sense viruses. And what is interesting is that you know, some of the ones that I've studied, piconaviruses like polio, flavi like Zika, toga viruses like alphavirus like chikungunya, their genome size are about you know, no more than basically 12 kb. But what is really interesting is that coronaviruses are off the chart here, which is way bigger than the other viruses. It's about 30 kb. And so you can imagine there could be a lot of problems with having such a large genome because of the mutation rate. And so we know that RNA viruses have very high mutation rate because of the error-prone RNA-dependent RNA polymerases. And so you can see here that this is a nice schematic showing you sort of the range of replication fidelity between RNA viruses versus cellular DNA replication machinery. And you can see here that RNA viruses have very towards the low fidelity end of the spectrum, whereas cellular DNA replication have lots of mechanisms that are in place to make sure that fidelity is maintained. And so meaning that there are not going to be a lot of mutations that are introduced each time when it's being, DNA is being replicated. What is interesting is that coronaviruses are sort of in the middle between general RNA viruses and cellular DNA replication. And the reason why is because it actually encodes an additional viral protein that I mentioned earlier, the exonuclease, NSP14, that has proofreading activity. And the reason why is because you can imagine, since coronaviruses are so much bigger than the other RNA viruses with the same high mutation rate of RNA virus, RNA-dependent RNA polymerase, coronaviruses could potentially reach sort of the error catastrophe very easily. And so you can imagine that the replication fidelity of RNA viruses is likely constrained to balance two things, right? So what I mentioned is that the virus need to maintain the genome stability so that it won't go extinct when it accumulates all these detrimental mutations. But at the same time, it needs to generate enough genetic diversity so it can conquer new tissues or new hosts, new niches. And so both are important. And the way coronavirus does it is that it basically encodes an additional component that can regulate its replication fidelity. And so what I want to end with is that here, looking at all the different RNA viruses and the kind of effects that mutations introduce into the virus that can change their replication fidelity, where mutations that will increase the fidelity, meaning that they are now less diverse, right, less able to generate mutations, versus mutations that decrease the fidelity, meaning that they're now more prone to errors. And you can see here that for all the other RNA viruses, such as preconaviruses and alphavirus, when you introduce these mutations into the RNA-dependent RNA polymerase that change the fidelity one way or the other, most of them you see turn out to be orange, meaning that the virus is now attenuated. So again, it's a very delicate balance how the virus, sort of want to fine tweak is replication fidelity, where too much or too little is both not good for the virus. What is really interesting is that for coronaviruses such as murine hepatitis virus, which is the murine coronavirus, and SARS coronavirus, you can see here that when they have lost their NSP14, the exonuclease gene, which then results in the loss of proofreading activity, these viruses have even more dramatic defect in their, basically, they're attenuated, and also a very dramatic reduction in the replication fidelity, suggesting that this exonuclease gene is absolutely critical for maintaining the fidelity of these viruses. And so I guess today there are a lot of topics to really cover, but I wanted to just sort of touch on, give you an overview of the replication strategy of the virus, and sort of touch on the different additional features that the virus has, and also interacting with the host, to give you a sort of somewhat complete picture of how these coronaviruses replicate. And that's it. I know I'm over time, and this is my lab and my collaborators and all the funding sources, but I really want to just end with this really funny meme. It's one of my favorites for the quarantine, is this baby that says, "Me after I eat all my quarantine snacks in one night." So thanks for your attention.
And then you have your children.
And super informative. What I'm going to ask is if people can please either raise their hand or if they can use the chat function to ask questions, so that you'll be able to get to them one at a time.
Okay, can we ask?
Yes, that's fine. I think that's fine. Go ahead.
Okay. So Melody, based on what you just were telling us about the proofreading function, it seems like a virus that had that gene deleted would be a very good vaccine.
Definitely. And people did explore that.
Uh-huh.
Yeah, no, that would be a great vaccine candidate. And Ralph Barrick's lab, I believe, at UNC, they did investigate that mutant specifically for SARS, and they did find that it's a great vaccine candidate. I don't know if anybody is working on that right now for SARS-CoV-2.
Because you could easily make a suppressing cell line probably that could supply the protein in trans, like what we have, what 293 cells are for adenovirus. And then you can mutate or delete the whole gene if you want, because it's complemented by expression of the protein from an integrated gene.
Right.
Okay.
Yes. Yeah, definitely. Yeah, and I think in the mouse model they tested for SARS, I believe it did trigger immune response, but it's attenuated. So you imagine that will be good properties for a vaccine candidate.
Mm-hmm.
Melody, I had a question too. I get to ask them all the time, so I should have asked this earlier. So I'm interested in the polyproteins. To what extent is the folding of the proteins or the assembly of replication complexes dependent on the initial translation of one big protein and then chopping it up? I mean, do they form a complex that needs that initial long protein?
Yeah, no, that's a really good question. I guess I think about like for some of the other RNA viruses, for example, flavivirus, you actually translate the polyprotein and immediately at the same time co-translationally, the protein is also being threaded into the membrane, right? Like let's say into the ER membrane. And then that gets processed. And so I imagine that also the orientation of the polyprotein is also critical for how these things get processed and how they interact with each other. So again, I guess getting at, it will be really hard perhaps to just express certain parts of the protein because you don't know if the polyprotein, if you absolutely require the polyprotein, some kind of conformation, right, to also trigger the next set of events. But I think for, you know, a lot of RNA viruses, I feel like it's all sort of happening, it's co-translational, and you sort of need the orientation in the membrane for all these proteins also to interact with each other.
So what is the chloroquine sensitive step for SARS-CoV-2?
I believe, so it's the acidification step, right? So I believe it will be the fusion between the viral membrane with the endosomal membrane. Correct me if I'm wrong. I think there are also other effects of the chloroquine.
And then so...
I think Jeff might want to add something.
No, that's okay, Arnie. Finish up.
No, I was just wondering, is there a range? I would imagine that you can get the endosomal pH to what you want it with by titrating in the right concentration of chloroquine. And is there a pH at which it's more toxic than not? So apparently you need that, for example, you need that pH change to present antigens to cells, to chop up proteins that are brought in by endocytosis, to present peptides on the class 1 and class 2 MHCs or HLAs, depending upon whether you're a mouse or a human.
Yeah, no, I see what you're like, you know, what you're getting at. If there is sort of a range or like a conditions, specific conditions that would make it only specific to inhibiting the virus, but not cellular processes.
Yeah, I mean, I guess malaria must be particularly sensitive since it's used, and it's used long-term in those patients.
Great.
Jeff,
Okay.
you want something?
I'll jump in. Melody, thanks for doing this. It was terrific. You know, given the sort of the characteristic of COVID-19 is this exuberant inflammatory cytokine response, which follows suppression of the innate type 1 and type 3 interferon responses, right? And you mentioned NSP1 and NSP3 as some of the immunomodulatory factors. Is that a druggable event? And should we be looking at inhibiting the suppression of innate immunity with these factors? It's kind of hard to imagine therapeutically, you know, after symptoms appear, how much effect that would have, given that, you know, the horse is already out of the barn by that time. But at least theoretically, is that something that people are looking at in terms of inhibiting those key interactions?
Yeah, no, that's a great question, Jeff. And I guess when I think about it, there's so many different layers to it, right? So I think, you know, the committee, we have had conversation about perhaps it would help, at least to help with the inflammation aspect of it. I think Doug raised this point, and Alex was also adding on, that you could treat patients that already have symptoms, because I think by then, obviously, it's too late, right, to use hydroxychloroquine or remdesivir, because the virus has already taken hold and the pathology comes from the inflammation, is to use immunosuppressive drugs, for example, right, to treat the symptoms of the inflammation. But in terms of the virus, you know, antagonizing the interferon, I think those will be great targets. So, for example, I mentioned with NSP3, I mean, it's so complex, right? So many different protein domains. There is this macro domain, which, you know, I'm very interested in, because we study antiviral PARPs, and the macro domain actually is now found from coronaviruses. Basically, it seems to have some effect antagonizing some of the PARPs, meaning that, you know, my colleague from Stan Perlman's lab, coronavirus lab, basically he looked at virus mutants that would have defective macrodomain activity. And what they found is that now, if you have PARPs, then the virus is defective. If you knock out the antiviral PARPs, the virus is fine, just like wild type. So I think macrodomain would clearly be a great target. And it's also found in a few different viruses, including alpha viruses. So it's really fascinating what these viruses are using these domains for. I don't know how druggable it is.
What is the antiviral PARP story? I'm not familiar with that. How does poly-A ribose polymerase inhibit the virus? Or
it's a great question. That's what everyone is trying to figure it out. We don't know.
What's getting modified, the viral RNA or the genomic DNA?
Potentially proteins, but it could be viral or host proteins. But we actually don't know. The only link we have is some of these viruses have macrodomain that removes that proximal ADP ribose. And there are PARPs that are antiviral, including the one we study called ZAP. But the direct link and also the substrates per se for ADP ribosylation is not clear. But definitely macrodomain will be a great target in responding to Jeff's question. But also, I guess the last layer, sorry, it's so long. The last layer I want to get is from my discussion with Bridget about the inflammation side of things, is that when you think about interferon, I mean, it's complex, because not all type 1 interferons are good or bad. Because when we think about, and I think Crystal can add to it, but when we think about type 1 interferon, you know, alpha is mostly antiviral, but beta is actually inflammatory and anti-proliferative. And so therefore, you can imagine that if the virus is triggering a particular type of type 1, like let's say the more detrimental type, that's not good. And maybe that's what's happening, but not
How
the
does
good
that
type,
work?
which is,
Don't they have the same receptor?
they do, but it's fascinating how there could be, for example, a binding affinity of type 1 interferon to the receptor could affect sort of the kinetics of gene expression. But that's a active area of investigation.
Mhm.
I could provide a small answer to that question in my talk in a couple of
Great, Alex, you come to my rescue.
Great. Looking forward to it.
in
What's your date?
Alex's talk,
What's
which
your
will be
date?
in
I'll
2
get
weeks.
it on my calendar.
So Melody, along those lines, if we think about patients and especially the demographic risk factors for patients getting severe disease, obviously it's older patients, and then things like obesity and diabetes and underlying lung disease. Are there known differences in sort of the interferon response in these patients? For example, in elderly patients, what's the interferon response like there? Is it muted, or is there sort of one that's more than the other?
Yeah, no, that's really interesting. I mean, I wonder if people have looked at, I mean, I know, like, for example, aging research, right? Probably inflammation, I would say, is more elevated in elderly individuals. And I think that is also telling you that it's not a specific genetic mutation, right? Like a predisposition. For example, in like STAT1 or interferon receptor or JAK kinase pathway, but more so related to age and inflammation. Yeah, it'll be interesting to see those studies in the COVID patients.
Great, thanks.
Yeah, thank you.
Yeah, we have a colleague, but I'm terrible at remembering names. But she's worked on the telomere decay in lymphocytes, and they go out in old people. Yep, you lose lymphocytes because of telomerase decay, telomere decay.
Interesting.
Great. Are there any other questions? So I guess if not, we have a remaining maybe 9 minutes or so, although we don't have to take all the time, but this is really, we wanted it to be a forum where people could make suggestions for what you'd like to hear in this seminar series, what you think would be helpful, if it's just in terms of resources that you think are needed on campus, or specific speakers that you think you'd like to hear from. We really want to keep this within UCLA because we feel like there's so much here, and we want everybody to know what everybody else is doing so that we can really set up collaborations. But I guess we want to know how we can be helpful to the community at UCLA who are doing COVID-19 research. So maybe I can just open up, and if anybody would like to chime in.
Well, I'm interested in what anybody wants to talk about. Thank you. It doesn't have to be about COVID-19, although it's a very interesting virus. I mean, most viruses are very interesting, right? The tiny ones are amazing too, with only 3 genes, 3 proteins. They do very well.
I agree with you, Arnie.
So maybe what I'll do is I'll just share my screen and I'll show you the ideas of topics that we had come up with and speakers, and then you can think about it, and then you can email me or Melody and let us know if there's other speakers that you'd like to hear from.
I'm enjoying the artwork behind you.
Oh, thank you.
I
don't know if this is if you can see my screen at all.
Oh, and also I want to make a pitch is that if you are on this list, you are among the topics that we think will be highly of high interest to this audience, at least for the initial round, before we get into all the COVID projects that are ongoing at the university right now. And so if you are on this list, please sign up for a talk with us. You should have gotten an email.
Yeah, and if you'd like to give a talk, please let us know, because we would love to include you on the list too.
And if you have other people that you think we're missing here, that will be great to give a talk on any aspects that are not included on this list. We like you to nominate them or have them email us. That would be great too.
Thanks very much. That was great.
Okay.
Yeah, I'd like to really thank Bridget and Melody for putting this together. It's a fantastic idea.
Yep.
Okay, well, thanks everybody. We'll see you
Thank you
next
so
week,
much.
Wednesday at noon. Okay.
Yeah, and there will be Bridget. I will be introducing her.
So it'll be a non-viral but very lung-related talk.
Which is also very relevant.
Thanks so much,
Thank
Melody.
you.
That was really wonderful. Thank you.
Great, thank you so much.
Thank you.
Thank you, everyone.
Thank
Thanks
you.
for all the questions.